Hi, everyone. My name is Nancy, and today I'll be talking to you about annotating genetic variants to target genes using high-C coupled magma, or H-MAGMA for short. So before I get started, I would just like to credit Doctors Christian and Danielle for developing magma gene set analysis. So magma is widely used in our field, and this is for various reasons, with a few being that the tool is pretty revolutionary in that it's aided in making sense of GWAS findings by identifying the target genes of genetic variants identified from GWAS. Additionally, magma is very easy to use in that you do not have to be computationally savvy to use the tool. Also, it is very efficient compared to other tools in the field, so it takes a very short time to run magma. However, despite all of these benefits, there are a few limitations to the tool. Namely, magma relies on positional mapping to link variants to target genes. We know from prior studies that the gene regulatory landscape is complex, and it's therefore possible for variants, especially non-coding variants, to interact with and regulate distal genes. So additionally, the regulatory landscape can be tissue or cell type specific, so we have to take into account the tissue or cell type that's most relevant to the trait or disease that we're interested in understanding. So these two limitations served as the foundation for developing high-C coupled magma, or H-MAGMA for short. So H-MAGMA complements magma by using high-C data. So as mentioned previously, H-MAGMA complements magma. So the materials needed to run H-MAGMA are the same as the ones needed to run magma, with the only difference being the gene to variant annotation file. So in this tutorial, I will walk you through how to generate the H-MAGMA specific annotation file, which links non-coding variants to their target genes using high-C interaction. So again, I will just walk you through how to generate the annotation file for H-MAGMA. So the materials needed to run H-MAGMA are similar to the ones needed to run magma, with the only difference being the annotation file. So to generate the H-MAGMA annotation file, you need a few other files. Namely, you need bed files for gene, exon, and promoter coordinates. Additionally, you need a high-C data sets in the bedp file format. You will also need an ancestry reference genome, and for the sake of this protocol, we'll be using the European ancestry. And once you have the annotation file generated, you need a GWAS summary statistics to run H-MAGMA. So again, the focus of this tutorial will be on steps A and B, which walks you through how to generate the H-MAGMA gene to variant annotation file. So I have broken the annotation files into 7 different steps with code for each step provided as part of this recording. So the first step is to load your libraries and all required data into a work directory. The next step is to read in your exon and promoter bed files and create genomic range objects for the exon and promoter using the exon and promoter coordinates from gene code version 26. Once you have this loaded and the G-range object generated, you will save them as an R file. So similarly, you would generate a genomic range object for all SNPs using the reference genome from the European ancestry. And you will also save this as an R file in the work directory for later use. So in the following step, we will overlap SNPs to genes starting with the exonic SNPs. So as a reminder, the exonic SNPs are assigned to their target genes using positional mapping. And this is because they are more likely to impact the genes in which they reside. So to assign the exonic SNPs to their target genes, we will overlap the gene range for objects for exon with the gene range objects for SNPs that was created in steps 2 and 3 respectively. So similarly, we will also overlap the G-range object for promoters with the G-range object that was created for the SNPs. So once you have both G-range objects created for the exons and the promoters, these will be saved as an R file for later use. So after we've assigned the exonic and promoter SNPs to their target genes, there will be a subset of SNPs that do not overlap with either. So these are known as the intergenic or the intronic SNPs. So in the following steps, I will walk you through how to match those SNPs to their target genes using Hi-C interaction. And this is shown here in this diagram. So to achieve this, we will first identify those intergenic and intronic SNPs and save them as an R file. Then we will load our Hi-C data. So for the sake of this tutorial, we'll be using Hi-C data from the adult brain as an example. So once you load the Hi-C data in the BEDP format, you will generate a G-range object for it. So we will next find SNPs that physically interact with exons and with these set of codes. Similarly, we will also find SNPs that physically interact with the promoter regions. So next, we will combine both G-range objects for the exons and the promoters to retrieve unique items. So lastly, we will overlap the intergenic and the intronic SNPs with Hi-C to identify their target genes. So once we have all these SNPs, namely the exonic, the promoter, the intergenic, and the intronic SNPs mapped to their target genes, the last few codes that I will show you in step 7 will be used to generate the annotation file that is compatible with MAGMA. So to achieve this, we will structure the annotation file so that it has the genes in the ensemble format, the chromosomal location, and followed by the SNPs that is assigned to each particular gene. So this is how you will have, so following these 7 steps, you will have your H-MAGMA variant to gene annotation file that can be used to run H-MAGMA for any trait or disease. So with this, you can run H-MAGMA for any trait. So for here, I'll use an example using Parkinson's disorder as an example. So this is the portion of the MAGMA file, the MAGMA code that you will place your H-MAGMA gene to variant annotation file. So here I am showing you one of the output files that you get from running H-MAGMA. So this is the .genes.out file. So with this file, you can actually extract the significant genes at various FDR thresholds for downstream analysis. So as I mentioned previously in the beginning of this tutorial, MAGMA has transformed the field of psychiatric genetics. And with H-MAGMA, we can address some of its limitations. However, it is important to note that there are still some pending limitations that aren't addressed using H-MAGMA, namely the directionality of effect. So even though H-MAGMA can assign SNPs to their target genes using Hi-C data, we do not know whether the SNPs, the variants are upregulating the gene or they are downregulating the gene. So to address this, you can use various EQTL data sets to analyze the directionality of effect. Additionally, we know that not all SNPs are actually implicated in a trait or disease. So you will need to do further functional validation to further prune down the list of genes. And then we also know that the sample size of a GWAS greatly impacts the number of target genes that is identifiable. So this H-MAGMA also suffers from this. And lastly, lack of diversity in genetic studies. Most of the analysis that we've done with H-MAGMA has mostly been from European ancestry. However, it's important to note that you can also run H-MAGMA for other ancestries. So with that, in the last slide here, I would just like to acknowledge where we currently are with H-MAGMA. So previously, we have generated H-MAGMA for brain tissues, including adult and fetal brain. We have also added various cell type specific annotation files, including cortical, iPSC-derived neurons and iPSC-derived astrocytes, as well as midbrain and dopaminergic neurons. However, H-MAGMA is more versatile than just brain-related cells. So to expand the tool, we have created additional annotation files for 28 different cells and tissue types to allow other researchers to use the tool. And these cell types are listed here, and all of these annotations files can be derived from our GitHub page. And with that, I would just like to thank my lab as well as my funding sources. Additionally, I'll be hosting a Q&A section on October 15, 2021 at 3:45 p.m. Eastern Standard Time. So if you have any questions or you would just like to say hi, please drop by to see and ask any questions. Thank you.